This is lecture 23 of Data Science 100 and Data Science 200. And the topic is clustering. So we're going to look at how we can use clustering algorithms to identify patterns in our data. So we're now at the end of the semester almost, and we've completed the core narrative arc of our class. We've looked at sampling. We've looked at the problem of tabular data manipulation and the solutions of pandas and SQL. We've looked at the problems of regression and classification and the solutions of linear models and decision trees. We've also looked at the problem of dimensionality reduction and the solution of principal components analysis and singular value decomposition. And it turns out that if you look at these last two problems, they're examples of machine learning algorithms. And these algorithms learn from data. So if we look at the taxonomy of machine learning algorithms and approaches, they fall into two broad categories. Those that work with labeled data for training purposes, and those that work with unlabeled data. So in terms of labeled data, that's supervised learning, where our goal is to create a function that maps inputs to outputs. And we learn our model we teach our model by giving it a set of training examples. These input-output pairs are labeled and consist of an input vector and an output value, that label. And we looked at two different types of supervised learning. Regression, where the output that we get out is a quantitative response, and classification, where the output that we get out is a categorical response. Now, the other form of machine learning is when we're working with unlabeled data, and that is unsupervised learning. In unsupervised learning, our goal is to identify patterns in data that does not have a label. We don't have a set of input-output pairs that we can use to train a model. So we looked at dimensionality reduction, where we had high-dimensional data, and we wanted to explore that data so that we could learn patterns. We wanted to perform exploratory data analysis. As humans, we don't work well with very, very high-dimensional data. We work best with two-dimensional data. And so we used the technique of principal components analysis to take our high-dimensional data, reduce it down to fewer dimensions, and then we could take and plot individual aspects of that data and learn something about our data. So the other problem is one of clustering. So let me describe to you what I mean by clustering. So here is an example. It's drawn from a figure that was on the fall 2019 second midterm. And the problem here that was on the exam was we created a synthetic data set of visitors to 8 different exhibits at a zoo. Then we took the principal components of that data. And so this is going to be linear combinations of how much time people spent at those 8 different zoo exhibits. So here's the first principal component on the x-axis, and the second principal component on the y-axis. And our goal in clustering is for each one of those points on our plot, each one of our patrons, we want to assign them to a cluster. This is an unsupervised task. There is no underlying label for each visitor. We just simply have the data, and we want to learn the label by just looking at that data. All right, so we don't have labels for each visitor, and we have to infer the patterns even though there are no labels. Just looking at the data, we have to see what we can learn. Well, if I look at this data and I needed to assign people to a cluster, well, it's actually, in this case, really easy, right? Because just looking at the data, I can see that there are 3 clusters. So I'll call one the yellow cluster, one the purple cluster, and one the red cluster. And then the points that fall into the yellow cluster will be labeled as yellow. The points that fall into the purple cluster will label purple, and the points that fall into the red cluster we will label red. Okay, so that's one really simple example using synthetic data. Let's look at a real-world example. So our first example is Netflix. So Netflix collects a lot of information about what you're watching, right? And using that information, what your viewing habits are, they could use clustering to do one of two things: to assign a person to a cluster and learn what kinds of movies does a given cluster, and look at what kind of movies does a given cluster watch. And they could assign shows to clusters. So two different ways that they can do clustering. Clustering of individuals, clustering of shows. And why would they want to do this? Well, they want to recommend to you what to watch next. They also want to have like categories of different kinds of movies that people seem to like to watch together, right? If I watch this movie, a lot of people will then watch some other movie. They could also use it for, they create lots of movies on their own. It's a very expensive process to create movies, and they want to know what type of movies should they be creating and look at how their movies do relative to other blockbusters and things like that. And so they can see, do their movies fall into the same clusters as the blockbusters, or are they in those like B movies that you only watch when there's nothing else to watch, right? So clustering, again, is different from classification, right? With classification, Netflix would be saying, this is the label in advance. So if we thought about movies, they would say, this is a sci-fi movie, this is an action adventure, this is a dramedy, this is a romance comedy, and so on. Instead, the clustering is going to discover the groups of our movies automatically, or similarly, the groups of our users automatically. So let's look at an example. So here are two examples. On the left, we have a cluster that Netflix discovered that contains movies like Scream 3 and the Dragon Tattoo trilogy of movies. And they've labeled this gory movies featuring a strong female lead. So the label is not something that they started with. The label is someone, a human, looking at that group of movies that was put together. What was a label that they could assign in terms of like a human readable label for people? Similarly, on the right, we have a cluster that includes Guardians of the Galaxy, Ghost in the Shell, and Jurassic World. And it's labeled blockbuster, visually striking, imaginative action. Okay. Yeah. That was clearly a categorical label that they assigned after they looked at all the different movies that were in that category. Again, the benefit for Netflix here is they can put labels that are interesting for someone who's I'm looking for a blockbuster that has lots of action, or a gory movie that has strong female leads in it. But they can also use it for their recommendation algorithm. So when they're clustering their users, they can see you like watching the movies that fall into the category of blockbuster, visually striking, imaginative action movies. And so they'll recommend to you those movies to watch next, and so on. And this is why you'll oftentimes see a movie that shows up in your recommended list that looks like nothing, like it's not a categorical movie that you might watch. Maybe you think you like watching action movies, and this is a drama or some other movie, but it turns out that people who like the same and watch the same types of movies that you do watch that movie, and they fall into the same cluster. So here's the second example of clustering that we can look at. So back in 2018, the instructors for 621B wanted to try and understand what are the factors that might influence student success in the course. And so they tried doing clustering by looking at the time and number of posts that a student made on Piazza, the time and number of submissions that student made to Gradescope, and the time and number of submissions that student made to GitHub. So that's basically when they saved their work. So what did the clustering algorithm discover? Well, it automatically identified those students that tend to procrastinate and start working on their assignments really late. So not really a particularly useful result, but still it's interesting that with no labels, the algorithm was able to determine that class of students. And then we can assign, after looking at that group of students, the label, these are the students who procrastinate on starting their assignments. So then you could think about, what could the instructors in 621B have done with that? Well, maybe they could change it so that the message of the day when you log into the hub is, for those students who fall into the procrastinating class, hey, maybe you should start working on your assignment early because this one's going to take extra time. And then they could do the experiment to see, did that actually shift the clusters around? Okay, so the third example I'd like to show you is one about taking and understanding biological processes. So here we have a plot that is of gene expression data. And so along the x-axis, we have columns that are genes. And the rows here are conditions. So it's something that we did to a cell, and it affected the genes in that cell. So in this case, the plot is it's gene expression, so it's plotting whether the gene is active and expressing, or it is inactive or turned off and not expressing. And it's a gradation, so that's why there's a color spectrum. And this is an old plot, and this plot I would not have created like this because it uses red and green colors. Green indicating that the gene is off, and red indicating that the gene is on. Unfortunately, there's a fraction of the population that is red-green colorblind. And so for them, this probably just looks like all the same color and is not very useful in terms of a plot. So always remember when you're plotting data visually, not to try and mix the colors red and green without differentiating them. So if you want to put red and green dots on a plot, then use dots maybe that are green and X's or pluses that are red so that people have two signals that they can use, shape and color, to distinguish between sets of points that they otherwise might not be able to. Okay, so a little bit of a digression, but again, what we have here is columns that are genes and rows where we've done something to the cells, like put a chemical on that cell, poured acid on the cells, and we see, does this cell respond by turning on the gene or turning off the gene, or partially turning it on or partially turning it off. Okay, and so what we can see by looking at this graph, so first of all, there's a huge amount of data in this table because there are a lot of conditions in terms of our x-axis, the rows, and there are a lot of cells that we have on our x-axis. And so we just tried looking at it, we would just see a bunch of green and red and whatever the color is in between blocks. What clustering does is group together cells and actions that are related. And so we can see, this is called a dendrogram plot, that there are two broad groups of cells. So along the x-axis, there's one group of, it looks like, not cells, genes, one group of 9 genes to the left. And in terms of conditions, we can also see that there are two large groups. There's one large group and one group of 6 experiments that are clustered together. And so we can see in the upper left, we have 9 genes that for the first 6 experiments were all turned off. And so based on what those genes are and understanding systems biology and the pathways and understanding what those 6 experiments were, we might be able to learn something that, or confirm something that we had as a hypothesis about how these cells operated or responded to the environment. So what clustering is doing, it's bringing these observations that are similar together and then presenting them to us so that we can try and understand or interpret what is going on behind the scenes. So here's another example of a dendrogram plot. This is for patients with a particular form of cancer called glioblastoma. That is a cancer of the brain. And along the x-axis here, we have 7 patients. And the y-axis here is genes. And so we can look at genes that are turned on or turned off, or at varying levels of expression for different patients with this form of cancer. And again, we can see with the patients that they fall into 2 broad clusters. Patients 18 and 19 on the right, we can see that the first set of genes is turned off. Then the next set is turned on, and then they're sort of decreasing as we go down. And then there's another block where they're mostly turned on, and then there's another block of genes where they're mostly turned off. And when we look at the other set of patients, we can see that it's actually kind of almost opposite which of the set of genes is turned on and turned off. Now, for those two patients, patients 18 and 19, they actually had increased mortality. And so one of the things that we might be able to take away from this is if we have a patient who's newly diagnosed with this form of cancer, we could sequence them, look at the gene expression, and if they fall into the clusters, the cluster with patients 18 and 19, then unfortunately we'd have to deliver some not so good news about the higher risk mortality that's associated with being in that cluster. This could also influence which particular drugs we use, or even influence which drugs we might develop based upon if we're trying to target genes that are associated with increased lethality.